The CFTR gene is located on chromosome number 7 at this particular position here. If we zoom in on this gene, we've got it just here. We can have a look at it a little bit closer. So it is encoded by 27 exons, these little bars here, which are the coding regions of the gene, meaning that they actually code for proteins or code for something. And then we've got 26 introns as well, which are these white spaces in between the exons. And these are sequences that don't actually code for anything. So if we have a look further at the exons without the introns, so we go through a process called splicing, which just cuts the introns out, we'll have a look at the mRNA of the CFTR, and we'll get the coding region. The coding region is just coding DNA. So no introns, just exons, and it is 6,132 base pairs in length. Now, when this CFTR mRNA undergoes translation, it produces a CFTR protein. And translation actually begins at this site here, indicated by the arrow. From this point to this point, it is converted into a protein and stops at the end here just before this 3' UTR, which isn't really important. It just is a non-coding region. It stops at a termination or stop codon. If I draw your attention to this label here, the CTT del, this indicates a deletion, which is also at this position here on the mRNA. And this deletion is only 3 nucleotides, so a C, a T, and a T. And when you look at that in comparison to the length of the coding region, 6,132 base pairs, it doesn't seem like much at all. But this tiny deletion actually causes a huge change in this mRNA and the way it is translated, and it causes a mutation in the protein and a defect in the protein's function, and hence causing a lot of the symptoms of the disease cystic fibrosis.